Direwolf de-extinction was the mystery, but it is reality no. So let's understand about how it can be de-extinction. So the species of canine de-extinction we will discuss in this video. So here is a white color direwolf shown in that picture. So in this way, in 2021, a DNA study found the direwolf to be highly divergent lineage when compared with the extant wolf-like canines. So basically, they contain a lot of the different type of species, but contain a canine. So that is why they are carnivores, and they was the predator of the human. So in this way, the first species named 1858 and extinction possibility is 12,500 years ago. So in this way, the domain is Eukaryota and kingdom Animalia, phylum Chordata, class Mammalia, and order carnivore. Basically, they eat meat, flesh of the other animal, and family Canidae, and subfamily Canini, and tribe Canini, and the sub tribe is Canina, and the genus Eocene Mariam 1918, and species Eocene A. dirus. So the characteristics we will understand is, so the characteristics of the dire wolf is the thick fur and the strong jaws as well as the broader skull. So the scientists collect the DNA from the bones which that preserved as a fossil. So the paleontologist discovered the fossil. So the definition of the fossil is the old remain of the animal buried in the ground. So this is the brown or gray wolf, which that used for the de-extinction we will understand. So here is the wolf jackal. It is Canis lupus present in this time, and this is also known as a grey wolf. This grey wolf important for the recombination. So here is the dire wolf which that fossilized. So the fossilization 13,000 to 17,000 years ago, and the freezing fossils can be occur in the cold weather, in the cold area. So the 14 genes we had extract by the restriction of the DNA sequence, but it is very difficult, remember, because the DNA is unable to preserve. So in this, the polymerization through PCR, the polymerase chain reaction to separate the 14 genes and amplification. In this, on the other hand, here is the grey wolf cell. Whenever we will grey wolf cells extract via skin, so in this way they contain chromosomes. So remember, and in this way we have target to insert the 14 genes to the grey wolf chromosome to become a recombinant. So the de-extinction required the genes editing, cloning, manipulation of ancient DNA, which that's come from the dire wolf. Scientists used CRISPR to edit 14 specific genes. If you don't know about the CRISPR gene, so you can watch my video. I have video. So in this way, here is the electroporation will lead to insert the 14 genes by the ligation due to the ligase enzyme used. In this way, the insertion of the new gene will lead to become a recombinant cell, which that is the origin cell of the grey wolf. So in this way, the recombinant cell of grey wolf will be processed to tissue culturing in vitro, and in vitro tissue culturing will lead to increase the number of the cell, which that is known as the cloning. So this tissue culturing is the cloning, but this number of the cell will increase only, but not become a zygote or something. So here is we will use the surrogate mother. Surrogate mother means the dog, female dog, which that provide the follicle stimulating hormone injection lead to overstimulation of the ovary, lead to primary follicle mature to secondary follicle and produce a lot more egg, and we will retrieve of eggs, and in this way the haploid egg which that retrieve and remove of nucleus. When removal of nucleus will lead to egg without nucleus contains cytoplasm, and it is have potential to become a fetus when apply the nucleus, which that's come from the gray wolf. So in this way here you can see this is the zygote, and in this way here is basically extraction of single recombinant cell of gray wolf and dire wolf combinant. So this is the recombinant mixture of the genes, 14 genes entry into the gray wolf cell. So in this way we will remove the cytoplasm while maintain the nucleus, and electroporation of other procedure to the zygote. Basically it is not zygote, it is only egg without nucleus. Will insert the nucleus, which that recombinant diploid nucleus, remember, which that able to divide. But the zygote will become a zygote, and the cytoplasm of the egg ability to divide and proliferation, differentiation to become a blastocyst in vitro. So this is the in vitro cloning will lead to blastocyst formation, and this is the recombinant cloning formation. And this blastocyst will insert to the surrogate mother, into the uterus of the surrogate mother to pregnant, and in this way the surrogate mother will be pregnant. Surrogate mother means whenever you will use the mother for cloning, but not their itself the children, so of suffering, so their own suffering, not but the recombinant and other type of the cell, desired cell. So this will lead to produce the two male born in October 2024 and female born in January 2025. So that is why it is very trending and hybrid character, gray wolf plus dire wolf. So we can get the characteristics and we can get the benefit from this white dire wolf, which that is very important and beautiful. So the lineage is very advanced, and in this way the dire wolf will de-extinction.